Research with Dr. John Kim and graduate student Alexandra Avra entitled Biomechanics of Glioblastoma Multiforme Organoids and the Extracellular Matrix. As a brief overview, we'll see that the purpose of this project is to develop glioblastoma multiforme organoids, GBM, and to test the relationship of these organoids to surrounding cells in the extracellular matrix, or the ECM. It also explores ECM development, variability, and reproducibility. So you're probably wondering what exactly is glioblastoma multiforme. Glioblastoma multiforme, or GBM, is a highly dangerous type of brain and spinal cord cancer. It's one of the main cancers in the central nervous system, and it actually accounts for 49.1% of all primary malignant brain tumors. GBM arises from a cancerous mutation in a type of brain cell known as the astrocyte. This is one of the supporting cells that is between the neurons, the neural cells in the brain tissue, and the blood vessels. It's in a very unique position where it's in a more complicated environment, something that will become important a little bit later in this presentation. Unfortunately for those diagnosed with GBM, it's estimated that more than 10,000 people in the US will die of GBM every year. As you can see from the graph on the left, even with the best supportive care we currently have available, patient prognosis is not good. The 5-year survival rate is only 6.8%, with the median length of survival being only around 12 to 15 months. And there's a couple reasons why treating GBM can be so incredibly difficult. The first is obviously going to be location. If you're having cancer in your brain, that's very quickly going to degrade other areas of your physical and cognitive ability. It's not like a cancer in another part of your body that you can just amputate or get rid of quite as easily. And some of the traditional methods to treat cancer, such as radiation or surgery, are going to be a lot less effective. Take, for example, doing surgery on GBM tumors. GBM doesn't form as like a singular mass. It forms and kind of spreads out into some of the surrounding tissue, meaning that if it was located in another area of your body, you could remove the tumor and the surrounding tissue to try to get all the cancer out. But with GBM being located in the brain, that becomes a lot more difficult to actually do. With radiation and surgery not being the most effective long-term solutions, patients are most often going to turn to chemo. The problem with this is that GBM is highly drug resistant, and the location makes it difficult to even treat with chemo drugs in the first place. Remember the location being between the neurons and the blood vessels. That means to even get cancer drugs to the site of the tumor, you have to cross something called the blood-brain barrier. This is essentially a very regulated filtration system that prevents unwanted molecules from even getting into the brain in the first place. And that unfortunately includes a lot of the current chemo drugs that we have. The current standard of care is temozolomide, which can actually cross the blood-brain barrier. But the thing is, even if you can get drugs like this to the tumor site, there's a lot of things about the tumor environment itself that make it very drug resistant and hard to treat. The number one part of that is heterogeneity. That is, you're trying to use one sort of chemical or one sort of drug to treat a highly complex and differentiated environment. Let's look at this a little closer. The GBM tumor microenvironment is not just one type of thing. It's not one type of cell. Instead, it's a very complex and heterogeneous mixture of different types of cells, different types of structures, different types of extracellular matrix with different biological components surrounding what's known as the necrotic core, which is a center of dead or dying cells. It's very complex, and therefore very, very hard to model. So the issue is when you have models that are trying to model something as complex as this microenvironment, it's going to be very hard to do. And that means that the research and the treatments are being based off of something that doesn't accurately reflect the type of microenvironment we see in real-life patients. So this kind of begs the question is, if we can better understand this microenvironment, the ECM, how it all relates, can we have better treatment plans in the future? We have a couple models that we currently use, the first of which being human tissue. This is just taking the tissue out of the human patient, studying it as it was in life. This is going to be the most accurate model, but have very limited availability. A second one that we use is animal models, and this is putting human cells into an animal and letting it grow there. The issue is obviously going to be if you're translation between different species, if you're trying to grow human tissue in, say, a rat, there's going to be issues in translation there. The most common and most broadly used is called a planar culture. This is most akin to the typical growing the cells in the kind of petri dish. The issue with this is even though it's available and widely used, it doesn't accurately reflect that microenvironment, that 3D architecture that was so important on the last slide. So that kind of begs the question, is there a middle ground? Is there something that's both widely available and that also can reflect the structure and architecture and components that we would see in real-life GBM tumors, and that's the GBM organoid. An organoid is a small, roughly spherical conglomerate of cells that actually more reflects the structure and components that we see in a real-life tumor. So how do we actually make these organoids? We get the patient cells, and then we get them as GBM stem cells. This is how they come to us. From there, we're going to grow them into something called a spheroid. This is just ensuring that we have a high density, high population of those stem cells that come from the cancer-mutated astrocytes. From there, they're going to grow into something called the full organoid, and we're going to put them in a small-scale bioreactor where they're going to go from the stem cell into that different mixture of cells through what's called transdifferentiation, becoming new different types of cells. And when we're done growing them over weeks or months, however long, however big we want them to be, they're going to look like these, for the example of the U251 and the JX6 stem cell lines that we actually use on this project. So what are we actually using these organoids for? The project is two-pronged. The first question is, do the organoids look and feel like the tissue of an actual GBM patient? Do they have the same type of physical properties? And the second question that my research more helps to answer is, do the organoids produce their own extracellular matrix? Together, these will show us how does the extracellular matrix actually relate to the structure and reproducibility of GBM organoids. So how exactly do I go about answering that second question? We mostly do this through something called immunohistochemistry. This is just taking the organoids and slicing them and staining them to look at different components of these organoids and of their extracellular matrix. This is going to show us different kinds of structures, different kinds of relative abundance. For example, if you look at the elastin stain on the far right, you can see clearly the necrotic core in the center. Because this was successful, we went on to also do fluorescent staining. The fluorescent staining allows us to look at both the cells and the components together. Take, for example, all the pictures on the left are of different stem cell markers, different components of the cells themselves, different components of the extracellular matrix, and we're able to overlay them and look at both parts of it together. Future work on this project mostly includes continued work for the immunohistochemistry staining and continued work on the fluorescent staining. Take, for example, if you look at the image on the right, we're not able to draw conclusions from that right now because one of the problems you see is that we don't have universal staining across the entire thing. As you can see, there's portions of it that are lighter, portions that are darker, and this is part of the things that we're going to have to continue to improve on this project so that we can make more conclusive judgments about it. Overall, we see that the whole purpose of this project is to explore the extracellular matrix, the biomechanics, to characterize the internal structure of these organoids, and see if they accurately compare to what we see in real-life GBM tumor patients. From this, we hope that we can make better models because we know that the general rule of anatomy is that structure does equal function. So if we're able to actually see the structure, if we're actually able to compare the very complex and very, very heterogeneous mixture that's actually inside a GBM tumor, then hopefully we can have a better model. With a better model comes better research, and with more accurate and more effective research, hopefully comes a better treatment for such a cruel and dangerous disease. Thank you. Are there any questions?
Hi, yeah, I was wondering, how new is this area of research? Like, is this something that's been previously studied at all?
So organoids in general are not one of the newest things. GBM organoids themselves have actually come up in the past couple of years. The novel thing about this project is how we're actually looking at the organoids. A lot of the research that's been done in the past has been about protocols for making organoids, and not so much about looking at the actual biomechanics, actually looking at the characterization, looking at the structure and components of it. So actually studying that part of the organoid versus just creating them or using them is part of what makes this project so unique and novel.
What do you find to be the most challenging part of this project?
I would say personally the most challenging part is that it takes a very, very long time to get from the stem cells to the spheroid to the organoids to the actual kinds of stains that you saw in the last couple slides. It's a very long process that can take weeks or months to do, meaning that if you make a mistake at the very beginning, you can see problems way down the end, or if you make problems towards the end of the whole process, you can ruin weeks or months of work. And because of that, it means that you have to do the most you can to meticulously follow all the protocols because it's such a long and very complicated process to get those kinds of images at the end.
To what extent does GBM usually spread?
So the spreading into the surrounding tissue that makes it very difficult to do surgery on and very difficult to treat. It's ranked from 1 to 4 on how aggressively it spreads. GBM actually is ranked as 4, so it's one of the most aggressive types of cancer. It doesn't actually spread mostly throughout other organs. It mostly stays in the central nervous system, in the brain tissue itself. It's not usually going to spread around, but within the brain tissue, it's going to spread or metastasize very aggressively throughout other parts of the tissue.
So I think around slide 7 or so, you described the process for how you grow the organoids, and you think you said it could take weeks or months. What percentage actually survive, or how, what's the efficiency of the viability of the obtaining the amount you would need for experiments?
So this project is not the one that actually came up with the protocol for how to do that. So by the time I'm actually working with the organoids and actually trying to grow them, the protocol has been a lot more perfected in that sense. Even so, I'd say that we have a pretty decent, we try not to lose them. We try not to have things go wrong just because they do take so long to grow. I would say most of the troubleshooting and the research on that particular part of the project came in some earlier steps that I was not involved in.